So good afternoon everyone. Welcome to Introduction to Proteogenomics Week 8 course for the live session for the NPTEL course Introduction to Proteogenomics for the week 8. So we will look at the different concepts that have been taught in this week 8, the different atlas of the human tissues that have been categorized and analyzed and have been put as a as a repository to all the scientific world. So we'll start from here. So first is I'd like to highlight is the draft map of the human proteome. So as we know that after the human genome map was established and after the human genome map was released, now the next thing was to look at that how the different genome is coded at the protein, and how the protein is how the protein looks for a whole human being. So similarly as the genome sequencing is done. So for looking at this, so this was the first paper which drafted the map of the human full proteome. So if we go and search for this paper, so draft map of the human proteome, so as you can see that the human after the human genome sequence, so this proteomic profiling of 30 histologically normal human samples was done, which included 17 adult tissues, 7 fetal tissues, and 6 purified primary hematopoietic cells that identified proteins encoded by around 17,000 to 94 genes. So around 22,000 genes are there present in the human body on an average, and among which total 84% of the total annotated protein coding genes could have been identified in humans using and was reported in this paper. And the main thing was that it was generated using it was done using a high quality mass spectrometry data set. So we will come into the next for the human protein atlas. So for the draft map of the human proteome was done using the mass spectrometry based data set. So here you can see that they used mass spec to generate the data and used FSEC human protein sequence database for identifying the proteins that have been identified. So if we go and look uh, uh, so if we go and look that the how it was done in the workflow, so in the workflow we can see that the adult tissues were taken, the proteins were extracted using the lysis buffer. So the lysis buffer composition you can find in the paper. So just remember that when we started in this approach, so first start comes the sample preparation, then it follows by the quality check using STS page, then followed by the trypsin digestion, then it is run in the mass spectrometry, and then using mass analyzer the M by Z ratios are found, and then from the M by Z different peptides are identified, and from the peptides are linked with the proteomic database, and this gives us the identification of the proteins. So here in this approach what they have done is that they have taken the different human adult tissues and the fetal tissues, and then they perform the same way, extracted the protein followed by the STS page gel to check the proteome profile of those tissues, and then followed by trypsin digestion, and then finally mass spec, and then finally data analyzed to identify the proteins of around 84% of the protein of the genes that encode for proteins could have been identified in this study. So moving on, if you have looked at here, so this helped in understanding of the landscape of this protein expression in all the cells and tissues. So this you can see that the expression levels and the comparison between fetal liver and adult liver. So fetal tissues and adult tissues could be done. They could detect the isoforms of the different proteins. So the proteins remains in different isoforms. So what are the different isoforms that are present? Then they could identify the novel protein coding regions in the human genome. So we are doing proteogenomics approach to understand and identify different novel protein coding regions, those regions that could not be identified, or those peptides or the proteins that cannot be identified only through the help of mass spectrometry, or using only a reference genome database, so which includes the proteogenomics approach. So they search this unmatched spectra using unique proteogenomics analysis. And by how they did it, said though they used one technique, that if the refsec transcript sequence is there, non-coding RNAs and pseudogens, so they translated it in silico, and then was searched against the theoretical protein amino terminity and predicted signal sequences, and they could translation of annotated, so they could identify around other exons, splice junctions, as well as novel protein coding regions. So they discovered around 808 novel annotations, including 140 pseudogenes, 44 novel ORS, 106 novel coding regions or exons within annotated gene structures, and 110 gene protein extension events, in addition to 198 novel protein cleavages. So they could, you can see that the protein database which was unmatched with the spectra with the refsec database. So refsec database is one which does not contain any redundant sequences. Okay, so which does not contain any redundant sequences. Now when the protein sequences were searched against those refsec, but there were some of the unmatched spectra, then 6-frame translation and the 3-frame refsec transcripts, pseudogenes 3-frame translation, 3-frame non-coding RNA, N-terminal sequence, signal peptides, when all these were clubbed, then it was identified, and then with further validation could confirm the presence of these proteins as a novel coding originated from the novel coding regions, or which has been coded from the pseudogenes or from the different regions. So genes whose exon products were detected. So here you can get a summary of all of the which it was identified. And if you go through this full paper, so you can identify different uh, different peptides identified, different power edges, different post translation modifications and all. So now this, they have documented this in human proteome map. So if you go and search in human proteome map, so here you can find that the human proteome map is there, which lists all their findings here. So you can, if you want to search for any protein and its expression, mass spectrometry based identification and its expression at a particular tissue. So for example, you want to find the basal level expression of GFAP gene. Okay, so you can select your pathways, select the gene family. So firstly, let us look an example. So if we go and WNT, okay, so you can see that the different genes and the different expression levels, which is not present in which cells, and here what is the low to high expression across the samples. So if we now look at the GFAP gene, GFAP, so we will just submit it. The GFAP gene, we can see that this highest level expression is in adult spinal cord. There are somewhere adult ovary, adult testes, fetal gut, fetal ovary. There are no expression levels, and there are medium to moderate in adult retina and adult frontal cortex. So now GFAP protein, if you find in your particular disease, if you are analyzing your data and find out that the GFAP protein or the GFAP gene is highly regulated, and it is not in the spinal cord but in some other tissues, might be in adult kidney or adult pancreas, then you can understand, and if you match it with this data, then you can understand this. Okay, so these are the basal level expression, and it should not be in highly expressed in adult kidney, but you are getting it, means that there is some disorder or something or the other is perturbed in that particular patient and or the particular person, and you need to analyze your data like this. So this another, I think in your course, in the videos you have about the HRG, histone rich glycosylation protein. So let us look at that, where does HRG's protein level expression is more. So you can see that HRG's protein level expression is more in monosaccharide. Others there are low to uh, low to medium level expression. So now in that video you might have seen that it is said that HRG is like a age related protein or age related gene, which is coming up as a marker, that it is as also associated with the death and other as a marker. So HRG's normal level expression should not be there. So if you find out in your sample that HRG's level is coming high when you do a shotgun or a lift tube study, then you might think that try to correlate that how it can be or what can be the reason for that. So you can download this in your JPG format or PDF format and can keep it, and also you can have a download data. So uh, if you want to download its full data, so you can do it by academic users or non-academic users. So you need to add your name, email, designation, institution, and which features of human proteome map will be useful to you, and I will not the share commercial purposes. And so if you want to download their data, so because if you want to query like this for each particular protein for particular tissues for each cell, then you need a whole lot of time. But if you want to download it and to automate your process, and so that you can analyze your data, match your data with it easily, so you can just fill up a form, register, and you can download as well. So this is where the human proteome map was done, and this was published in 2014, and it has been set up as a web portal where you can identify the expression levels of the mass spectrometry based identification of the different proteins. So if you want to have much more answer to their questions, so now you can see that what is a human proteome map portal. So human proteome map portal is a web-based resource that reorganize mass spectrometry based proteomics data. So here you need to understand that the human protein atlas that I'll come to next, and what was explained in the lectures, that the human protein atlas was done on the basis of the RNA sequence expression data and the microarray based tissue microarray based data, which dependent on the antibody. So in this both studies, that major difference is, one is the antibody based method, another is done the antibody free method. So so you can just understand that can also correlate the difference of these both techniques in identification of the expression of the protein level in human tissues. So all these data from individuals with any known diseases were obtained from histologically normal samples. So all this data has been drafted from the histologically known normal samples, so that you can get an idea that okay, so these are the basal level expression of any tissue for a particular protein or the particular genes. So these are expressed at the basal level. So any upregulation or downregulation might indicate that it is some that there is some disease in that person, or might be indicator as a biomarker, or can be as a prognostic marker, or can be the cause of the disease, or can be the effect of the disease. So all these protein markers that are identified generally can be they are increased due to the cause of some of the upstream or downstream effects that increases that level, or because of their certain increase in their expression level causes the disease. So this can be both cause as well as effect. The biomarkers can be both cause and effect. So this gives an excellent idea of what the basal level expressions are, and what basal level expression should be, and what from where you can get a reference of their upregulation and the downregulation. So what is the quality of the data? So all data were acquired on high resolution Fourier transform mastery using SCD method for fragmentation. So if someone asks the different the quality, how was the data quality? So So they have mentioned the HCD fragmentation using the Fourier mass spectrometers. So how are the expression values calculated? Spectral counts per gene per experiment, where first summed from all peptides mapped to each gene. Total acquired tandem mass spectra were used to normalize between experiments, and then spectral counts per gene were averaged across multiple. For example, pancreas. Final average spectral counts per gene per sample were used to plot the white to red gradient, while sound spectral counts per peptide were used to depict the heat map for identifying mapped of interest. The expression values that were calculated were done using spectral counts per gene per experiment. So both the experiments of the SDS page and the uh, RPLC reverse phase based fractionation. So both the experiments average spectral counts were taken and normalized between experiments, and then were averaged across multiple experiments to confirm their chat. So firstly, there were two columns, SDS page and the RPLC fractionation. So from there the spectral counts per genes that were mapped were taken, then they were normalized and then were averaged across the experiments, and then they were calculated on the basis of the expression, that how it can be, the expression can look like. So how is the expression level of a gene is on the basis of all the peptides sorry, mapped to the given gene as a result of a query, and the abundance of peptides is displayed on the specificity at the gene level. So how do we download mass spectrometry? So you can, as you know, that the Proteome Exchange consortium, or the Proteome Exchange gives you, is a full repository of the proteomics data, the PRIDE. So generally the raw files that are generated from mass spectrometry based data are uploaded there, so that the data is available to all the reviewers as well as the researchers worldwide, so that they can easily download your data and can reanalyze your data. So if you want to have a particular, so if you are working on a brain disease or a liver disease, and if you don't have a normal liver data and you want to compare, you have only the disease data, so you can download these raw files and can analyze accordingly, and can identify the differential expression of proteins in that particular disease. So if you have samples of some rare liver diseases and don't have the normal liver samples, so you can download this data and then can easily do a match and can identify and say that these are the proteins that can be taken as a marker for the prognostic or the biomarkers. So this is data and how to cite. So this is the overall for the human proteome map. So now looking at the tissue based map of the human proteome, which was another at near about the same time, which was published to identify the what is the protein expression levels, what is the gene expression levels across the tissues. Tissue based draft map of the human proteome. So it was on 2014 and it is in 2015. So 2015. So protein expression across human tissues. So as we know that the sequencing of the human genome gave very good new insights into human biology and disease. So dynamic expression of each of the approximately 20,000 protein coding genes, so now present across 32 human tissues. So they took around 32 different human tissues. So they not only measured expression at an RNA level, so majorly what they did is to take the expression at an RNA level, that how the transcriptomic sequence are coming and how they are generally being expressed across the tissues. But also what that been done, also used antibody profiling to precisely localize the corresponding proteins. Okay, so the thing here, the difference between a draft map of the human proteome and the tissue based draft map of the human proteome, the two major difference was that one used fully mass spectrometer based approach, and the other used antibody based and the RNA expression level based. So antibody based rely on, one is on the antibody free method and one is the antibody based method. So there are pros and cons are both there for both the techniques. For example, at the antibody based methods, you need to have the expression of those unique proteins, then you have to generate the antibodies, which is more cost dependent as well as the time dependent, and you have to purify those antibodies, and then only you can perform the experiments. Whereas in the mass spectrometry based approaches, so you need to just have the samples, do a sample preparation, and then can analyze those samples that which proteins are present. So but also antibody based methods are more specific, and you know that your antibody will bind to a particular specific protein of interest, and you can be very much sure about uh, you must be very much sure about how much your data is being uh, shown, and how much your protein is very much confident of the protein expression or the signal that you get due to the antibody based method. So these are the different pros and cons of both the methods. So now if you look at there, so this was the final conclusion of the brain, the endocrine tissue, lung, blood and immune system, liver, male tissue, adipose tissue. So these are the human tissue enriched proteins, and they also went ahead and filled the membrane and secreted isoforms. So then they have created a comprehensive all their data in the human protein atlas. So we will go. So we have determined the number of genes for which evidence is level at protein level by antibody based data with manual annotation of the literature by Uniprot consortium. So they have combined their antibody based data with the manual annotation of literature of the Uniprot consortium and the results from recent mass spectrometry based proteogenomics analysis. So there are 17,132 protein coding genes with proteins identified from at least one of the three efforts. So there are 17,132 protein coding genes which have been identified at least from the transcript level. So what are those three efforts? One is at the transcript level or RNA sequence level, one at the antibody based level, or one at the mass spectrometry based level. And 13,841 genes with the experimental evidence from at least two of the efforts. So among the three efforts of the microarray using the antibody based approach, or the mass spectrometry based approach, or your what you say your uh, uh, sorry the other one approach is the RNA transcript level approach. So at least two of the efforts we got the 1384 genes. Further, there is evidence at the RNA level. So only when there is an evidence for the RNA level or the transcript level is for the 2546 additional genes within either our data or annotations by Uniprot. Although proteins not yet detected by one of the three methods should be further investigated to establish them as true human proteins, it is noteworthy that out of the 20,356, so among the 20,356 putative protein coding genes, there are only 677 genes of which there is no experimental evidence. So among the 20,356 genes, there are 677 genes for which there is no experimental evidence from all the three efforts, like antibody based method, tissue based method, or your transcript level, and others non-coding genes. Thus, it is possible that most of these missing genes will be removed from in the future. Only at the RNA obvious targets for in-depth functional protein studies. So the genes and the genes with evidence only at the transcript level. So what those genes we have got evidence at the transcript level, but could not be identified at the proteome level with both antibody-based approaches or mass spectrometry-based approaches should be obvious targets for understanding of their functional protein studies. So they can find out the summary of the supporting data. So, and what are the different number of genes, fraction of transcripts, and those tissues. So the tissue elevated proteome, number of tissue enriched genes. So there are some genes that are enriched for a particular tissue. So you know that the differential splicing or the spliceosome helps in the differential expression at the different cells, and which helps in functioning of the different things by different regions. In the human secretome and the membrane proteome and the housekeeping proteome. So this is very much important as to understand that the most that all the cells contain a particular number of genes which are housekeeping in nature, which does not change with the tissue level or the on the context of the tissues or the function, because this helps in the cells generation, cells proliferation, cells maintenance, membrane integration. All these are the housekeeping proteomes. So you can get to know that if you target, if these housekeeping proteomes are altered, then it can cause us much more difficulty. And the regulatory proteome, the transcription factors, and the druggable proteome and the cancer proteome, I think this has all been discussed in depth in the videos for this week's session. So I just wanted to give a glimpse, an idea, or that that there have been three efforts altogether to understand the proteins, to identify the proteins that are present across uh, across. Yeah, hello. So actually, I have started the session, and now I was discussing about the tissue-based map of the human proteome. So the thing is that so there I will just repeat it once. So if I say that first effort would have was the draft map of the human proteome. So in the draft map of the human proteome, the it would identify the proteins expression using mass spectrometry based approaches. And then the human protein atlas, which is the tissue based map of the human proteome, which was published. So they used two different techniques. One was the RNA sequence level using RNA sequence, and one was the antibody based method using tissue microarray expression. So these are the three different efforts that have been taken place to identify the human proteins at a stretch, or the identify the proteins of all the protein coding genes. So so among the 20,000 something genes, it is mentioned here. So you can identify there was around 17,000. One second. So around 17,000. Yeah. So among the 20,356 putative protein coding genes, there are only 677 genes for which there is no experimental evidence. So are only 677 genes. So all the three efforts, mass spectrometry based, RNA sequence based, or antibody based, there is no such genes. Okay, so these are the missing genes, and there are 2546, around 2546 genes that have evidence at transcript level but no evidence at the protein level by both antibody based method or the mass spectrometry based method. So these are the genes are very much useful for targeting, for understanding, for their different proteome studies. So these are the very much interesting targets to look after. And the different proteomes, like the tissue elevated proteome for the tissue enriched regions due to the differential splicing of the cells, or the human secretome, or the membrane proteome, or the housekeeping proteome. So housekeeping proteome, so percentage of genes are present in all the cells, which helps in their proliferation, their growth, maintenance, and the integrity, membrane integrity. So these are very much important important genes. So this needs to be kept intact, and their enhanced or decreased expression might lead to much more uh, fatal causes. So these all have been, I think, discussed very much in the uh, in the video for the druggable proteome and the cancer proteome and the tissue versus cell line isoform proteome. So the new thing which I just mentioned was the draft map of the human proteome. So this was came around in 2014, and the tissue-based map of the human proteome came in 2015. So here they used 17 adult tissues, where they have used 32 tissues and the different 7 fetal as well as the cells, and they could identify around 84% of the total annotated protein coding regions. So human proteome map looks like this. So if you search it, so I'll just place the in the chat box. So if you go into the human proteome map, and if you go for the FAQs, so you can read about it, that what is a human proteome map portal and how they have mapped. So there I just explained it that they use two different things using SDS page and the RPLC fractionation, and then they identify the spectral counts per gene per experiment, and then identify those, normalize those within experiments, and then the spectral counts per gene were averaged across multiple experiments to identify that what are the differential expression. So here you can go for downloading. So if you want to download, so you can, as an academic users or non-academic user, you can you can register and then can download your data for if you want to have a query. So the here what is the problem is that, the benefit is that, for example, you are working with a liver disease and you don't have a normal liver sample, so you can just download their raw data of the normal, because these are from the patients which are fully normal, histologically normal. So you can just download the data and then can analyze in your own way, and when you analyze it in your own way, you can identify those proteins or the markers which are differentially expressed. So now if you, for example, if you search a gene like GFAP gene, and you want to identify, because in your kidney you were, for example, you were working with kidney proteome and you found out that GFE expression is way too high, and you try to identify that if their adult kidneys, what is the basal expression level in the normal tissue. So you can see that it is not so highly expressed, but if you find out in kidney that GFAP protein or gene in your data is highly expressed, then you will definitely go and search for the that why it is highly expressed, and what is the reason for that. So this helps in understanding for it. So now is the human protein atlas. If we go and just give an overview of the website is, so here it is written different tissue, brain, single cell type, tissue cell type, in fact metabolic. So if I just go with a metabolic subsystems, cellular composition, so if you go for the beta oxidation of branch chain, so you can see that in the pathology, so what are the enzymes, tissue enhanced, detected in all, protein evidence, pathways. So you can identify everything, and so subcellular localization or subcellular map of the human protein. So what I would like to show is that if you want to download your data, so you can go to downloadable data and you can hear from here. So if you have a huge file and you want to analyze, so you cannot go and search one by one. So you can, the best way thing of these things are that you can download the data, you can analyze your data, you can refer to your data with these repositories, and then you can come to conclusion. So pathology data, so all this you can get in the TSV format. Subcellular localization data, then if you have a different proteins list or the genes list, and there you want to identify that they are present in which subcellular localization, just you need to download it and then identify those. Then RNA construct tissue gene data, so you can download all these genes, pig brain, mouse brain, all the data they have. So you can see that how comprehensive the database are, and you can download and identify those. So now let us search for GFAP only. See GFAP, so here we can see that it has tissue enhanced, pathology enhanced, single cell type, brain supported, subcellular, and cell line. In cell line where highest expression is U87 mg, and metabolomic data is not available. So which one is not available will you show is not available, and the rest you can see that what has been reported. So if you go for the pathology, so here you can see that protein class, predicted location intracellular, number of transcripts, and it you can see that HPA cell line. So which cancer category it is highest, you can see glioma. So you can, from here only you can identify that your particular genes. You can see that in any other cancer, thyroid cancer, lung cancer, colorectal cancer, head and neck cancer, in any cancer it is not written, it is not there, but in uh, glioma it is highly expressed. So it is for sure that in the glioma or the brain tumor of the glial cells, you will get an high expression of GFAP protein. So GFAP protein is a marker, you can tell in a way that it is a marker for the glioma tissues. From the pathology atlas you can easily get. So now if you go for the tissue, so now just see the difference, that here if we go for the human proteome map GFAP, where it has shown and where it is showing here. So RNA expression in TPM, so brain is showing the highest. Okay, so expression detection. So, and what is the expression level of the fetal brain and shattered spinal cord and this brain? So it is showing that it is the highest expression, and in eye also you can see that there has been some expression. And so easily you can say that this is present in brain, and in the glioma, one of the kind of brain tumors, it is again highly expressed. So it is basal level expression is there, plus it is also a positive marker in the some drug, the cancer like tumors like glioma. So this is the whole human protein atlas, where is the protein expression score based on antibody base and the RNA expression base. So this is very comprehensive data set, both which can help you in understanding. Now one thing is that there you can see that it is written in RPKM and FPKM. So I thought just giving you the definition, what is the meaning of RPKM and what is the meaning of FPKM, so that you can understand that what does it defines or what does it intend to mean. So RPKM is reads per kilobase of exon per million reads ripped. So this helps to understand the compare the gene expression levels. So you can say that it is the unit of the gene expression levels, uh, and FPKM stands for fragments per kilobase, that is reads per kilobase of exon per million mapped reads. So it is more or less analogous to RPKM, and it is specifically in paired and RNA-seq experiments. So RNA, the sequencing experiments can be both one end or paired end. So for the paired end, it starts from both 5' ends and the 3' ends to sequence your data. So FPKM is generally used for this paired end sequencing, and whereas RPKM is used for the single end sequencing. So reads per kilobase of exon or fragments per kilobase of exon per million map reads. So what is that? So these are the raw reads or fragment counts. LI is the feature, that is gene or transcript length, and QJ corresponds to the total number of mapped reads or fragments. So total number of mapped reads or fragments, when it is multiplied into 10 to the power 9, so it on the basis of per million, it gives that how many reads have been mapped to that particular fragment, and which compares that, okay, so these are the gene expression levels. So from where gene expressions of both can be compared. So this is the meaning of RPKM or the FPKM, and this is an unit of comparing data. So this is the next is that another important is that atlas antibodies. So all those antibodies that you have that they have been used for identification in the human protein is available, and you can find them and you can order them for your experiments if you want. So to go for the atlas antibodies, so if you want to validate, or if you want to see that for your particular disease, for example, if you are working with some rare diseases, that how that particular protein which is like GFAP is enriched in brain, you are working with a rare brain, how GFAP you know that it is enhanced in glioma. Now what happens to this GFAP in some rare neurodegenerative diseases? So cancer is one where it proliferates, cells proliferates, and in neurodegenerative diseases what happens, that the cells death happens or cells dies. So whether GFAP is also reducing, whether GFAP is helping in proliferation, whether GFAP is or level comes down when the neurodegenerative diseases happen. So that you can try to verify with this and plus antibodies. So you can find these antibodies, you can order them, you can perform the experiments, and then you can conclude that okay, like it has been shown that GFAP helps in enhancement for a particular diseases. Now again you can check and see that okay, no, GFAP in neurodegenerative diseases has also a role or has a different effect. So for which this atlas antibodies are also very much helpful, and also you can know. So these are the different repositories that can helpful. So now I just want to give this analogy is that I like this analogy personally, is that you know that globe was present from many times, but just try to understand that if someone has been handed over a globe and you have been asked to go to a particular location, so it was not very fruitful, right? So it was not in the correct form which could have been useful for a layman or for any person, to be specific, not to be specific, for any person to go from this region to this region. But with the induction of the Google Map and with the presence of Google Map, now each person can go and okay, can go to any location and can easily pick up a set a location and can go easily without having any effect, without having any worry. So the both the things, both the globe and the Google Map is doing the same thing or showing the same thing, that is the locations and the maps and the location of the countries or the places, but there is a huge difference in between because of its form. So if someone is doing just identifying the protein data and is not keeping it in a repository or in a way or format where layman can go and use those data, so where a layman cannot go and use those data and can refer to his or her own data and can analyze and can try to build up or make up something, uh, then it is of no use. So with the presence of this human protein atlas or the human proteome map or the antibody atlas as I showed, so this can be referred to as, can be analogous to what Google Map has helping us in day-to-day daily life. So the researchers are also getting benefited from this atlas and all these expression levels in understanding the diseases and can to help and treat the patients better or to make better treatments available. So this was means I personally like this analogy way bit to, so that to understand the importance of all these studies and importance of this generating this type of data to analyze this type of big data and how it is helping millions of lives. So now the next thing is that, so we so in every week actually we had given this tool, but this week IGV or integrative genomics viewer was thoroughly discussed in the videos, which need to be understand the different approaches used to it. So I thought of just going through its user guide. So it is just an easy tool just for mapping and the for mapping the sequences and to visualize the alignment sequence. So it can be easily done by downloading it. So the down IGVs can be downloaded if you want, but you need a BCF file, and that needs time. Actually I've shown one day in Galaxy how you can do it. So if you find any reference data from which you can do and then can look at it, IGV how it functions. So if we go at IGV, so they have a very extensive manual. So if you go for the documents IGV user guide, so you will come across this kind of interface where there is written about the IGV user guide. So user interface. So now next we can go and navigating the view. So here in this lecture, in the lecture has already given you some of the hands-on session where they have asked, where it is mentioned that how you can perform the visualize the alignment. So here you can check that you can zoom it and you can look at the chromosome wise. So major thing, important thing is to look at your data chromosome wise. If you are interested for a particular chromosomes, and you can go for a particular chromosome wise, otherwise you can easily go to different chromosomes and look for a whole data. So you can zoom and you can select your chromosome. So if, how to zoom in, how to zoom out, so I thought of just giving this reference so that you can if you are doing any day and you are stuck in somewhere, so that you can just go to their user guide and can do it on your own. So double click or shift click and the scroll bar. Okay, so this is for the loading those genomes, then viewing the data and changing the display. Okay, so you can visualize it as a heat map, bar chart, scatter plot, line plot, as you wish. So okay, so this is a graph type, data range, track line, color region. So this gives you huge things to do. Then if you have the segmented data, now major thing is for the viewing alignments and interpreting the color. So what is the interpreting color by the insert side? So so when we are mapping, so we will be generally looking for what is not getting matched, what are the mismatches present, or what is getting inserted or deleted or getting additional in the genome. So that's where comes the major important goal. Okay, so the IGV uses color coding to flag anomalous insert sizes, so which are not normal. Okay, so whichever not normal insertions or deletions are there, so IGV uses a color code so that a person can understand that what is the thing. So this color code needs to be understood to interpret those. So when you select color alignments by insert size, the default coloring scheme. So if you don't change the coloring scheme, this will be the default coloring scheme. And now if you want to change, okay, so now I want to change my uh, coloring uh, coloring scheme and to give any specific color, so then that is a different issue. So for inferred, that is larger than expected, possible evidence of a deletion. So if there is an evidence of a deletion, then the default color scheme is red. The default color scheme is red when there is a chance of possible deletion. When it is insertion, then it is blue. And by the chromosome, these are the different colors for the chromosome code 123 XY. So 1 to 22 and the XY, these are the different color codes for chromosomes, and blue for the insertion, red for a deletion. So these are the default color schemes. If you don't change, and if you change, then it would be definitely would be different. Then deletions, reference genome, subject genome, there will be a difference, inferred insert size and expected insert size. So you can see that red, you can if you load your data, and then you can find out that there are red from here. So you can see that there are deletions are there. And when you can see that there are blue, then there is an insertions. And interchromosomal range, so interchromosomal rearrangements can be done. So there also like this which interchromosomal arrangements have been done, then it gives a specific color code. Okay, so this is the alignment. So from here, if I just see, I think you can find out that it is shown in the, it has been shown in the video as well. If you show the bases, so either you can look for a particular base for a particular base, or if you click on right click and give that, okay, I want to check for all the bases, then you can, all the bases will be visible, and then you can find the sequences that which one is matching with your reference genome or the normal genome, and which one is not matching with the reference genome. So this is the total count and the number of A, number of C, T, and which are the matches and which are the mismatches. Everything is been done. So another thing is that I think if you remember that you can get the gray colors and the white color, so depending on the mapping quality. So if it's mapped qualities for a particular range, then you will get uh, the gray color much deeper or darker, or vice versa. So this is for the IGB, and also sufficiently line at the center of the display. So alignment track, the center line becomes two line, detecting structural variance, mapped reads, color and transparency for mapped reads, insertions, deletions, coloring and sorting, view as pairs, okay, coloring of mapped pair reads. So actually this is a very extensive thing, and the software is very easy to use, but it's also very much extensive in understanding. It helps you understand every part of the alignment of the sequences that you have given, so you need to go through it thoroughly, and if with one parameters change set, you can easily understand what you are looking for. So they have given an extensive guide, so interpreting color by bisulfite mode. So if there are CPG islands and all, then viewing variance, multi-locus view. So if you want to identify using multiple loci, okay, so for these particular genes, so for example cake colorectal cancer, they are take care as MIG, so you can easily identify it. Then the regions of interest, so if you just go through it, you can identify and understand that how much more you can know. So the mostly the color code, color schemes, and how to look at the bases and the base pairs, and from there only you can try to identify that what would be the novel protein coding genes, as shown in the video, in the lecture of the IGB hands-on session, that you can identify that protein coding regions, and then what would be the novel proteins. But there are software like custom prod BJ quilts, etc., which helps, which by giving the VCF file, they will give you the protein coding regions of the genes, that will be the novel transcript region that you can search in mass spec and can identify the novel protein regions. So this was all regarding the this week's lecture for the human protein atlas, human proteome map, antibody based methods, mass spectrometry based methods, their expression levels, and the IGV or integrated genome viewer and their application. So are there any doubts or anything to ask? The rest I had said in the previous, I started the lecture, so you can look at the YouTube video as well if there, if you have missed anything, or are there any doubt, or I'll end the session for today. Okay, so I think so we can end the session for today. So this was for week 8 of the introduction to NPTEL course, Introduction to Proteogenomics live session. So thank you all for joining.